In our lesson on blastocyst development, we had a brief look or touched on the concept of stem cells, but in today's lesson we're really going to dive down deep into what it means for cells to differentiate from those original stem cells and what are some of the different cells end up becoming. So to start us off, let's define what cell differentiation is. So very loosely, this is when a stem cell becomes specialized. So we can see in this diagram on the right hand side cell differentiation and specialization in process, where we start with a stem cell and this gradually becomes a more specific or a myeloid stem cell. This myeloid stem cell then becomes a myeloid blast, which can then develop into things like red blood cells, platelets, or even white blood cells. Now what you'll notice here with white blood cells in particular is that there's not only one stem cell which develops into white blood cells. So for instance, a stem cell can also differentiate into a lymphoid stem cell, which can differentiate into a lymphoid blast, and lastly this can develop into white blood cells. So to make it really easy and to give you a simple example of what cell differentiation can mean, this is where a bone marrow stem cell becomes a red blood cell. Very simple example. So when we talk about specialized cells, specialized cells are anything with a specific structure and function. Stem cells don't have that. They don't have a specific structure and function. They're not ready to go. When we look at a specialized cell here, so in the top left we have a sex cell, over on the top right we have a fat cell, and in the middle down the bottom we have a nerve cell or a neuron. These three distinct types of cells have very different structures, very different functions. So, stem cells, they themselves are not specialized, but they are capable of mitosis. Now, given the right conditions and very specific conditions, they can then differentiate into specialized cells. Now in the human body, there are 200 or roughly 200 different types of cells that make up our body, and they all come from the stem cells of the inner cell mass of the blastocyst. And this is what we've spoken about previously, right? So we've previously spoken about the stem cells and the inner cell mass, and we've spoken about how they will end up developing into what is found in the human body. So this here is the blastocyst, and to quickly recap, this inner cell mass is this lump of cells contained within the cell itself. Now, when we're talking about what's in blue here, this blue layer is known as the trophoblast, and this is what is one day going to develop into the cells of the placenta. The inner cell mass, on the other hand, is going to develop into the cells of the fetus, or into, one day, the adult. So when we have a look here, there are three distinct levels to stem cells. Level 1 is a totipotent stem cell. And these totipotent stem cells can form not only the embryo, but also the membranes or embryonic membranes that surround, support, and nourish it. These cells occur before the formation of the inner cell mass. The next one is what we've just spoken about, which are pluripotent stem cells. They are capable of giving rise to not all of the cells in an individual, but they're not capable of giving rise to all of the cells that will develop or help to develop the organism, but they are capable of developing all of the tissues of the organism, just not those membranes or embryonic membranes that are going to surround, support, and nourish them. Lastly are adult or multipotent stem cells. And these can only give rise to cells that have a specific function. So another example would be our bone marrow cell, giving rise to a red blood cell, same as the example that we spoke about just a moment ago. So in terms of these totipotent stem cells, once again, to reiterate and be really, really clear, they can differentiate into all cells. That includes that embryo and the membranes, those embryonic membranes surrounding it. Now, these cells in particular are found in the zygote before the formation of the inner cell mass. But after a period of about 4 to 5 days, as that blastocyst develops, those totipotent stem cells begin to specialize and form that blastocyst or blastocyte. As those cells have differentiated into the blastocyst, that outer layer of cells is eventually going to form the placenta. And this outer layer of cells, I'll highlight this in blue so that it lines up nicely with the trophoblast down the bottom, come from that trophoblast. So when we talk about what that layer of cells is, we're going to name it as the trophoblast. On the other side of things, if I do it in a light pink for the inner cell mass, this is going to form all of the tissues of the human body. And these stem cells are pluripotent. This means, again, that they can give rise to all of the cells found in the organism, but not necessarily, or not those, embryonic membranes. So, embryonic stem cells, therefore, are pluripotent. Multipotent stem cells, or more simply, adult stem cells, are those that are multipotent. And these multipotent stem cells give rise to specialized cells. So again, the example we've used twice already is that bone marrow can give rise to red blood cells. Okay. So let's now talk about what happens to these multipotent stem cells following implantation. So following on from our last lesson. So during implantation, at around the 3rd week of development, the inner cell mass cells become multipotent. And this leads to what is known as the embryonic germ layers, or the primary germ layers. And there are 3 of them. Each layer differentiates into different parts of the body itself. So the first layer is known as the endo, ecto, and mesoderm. Not the first layer, each of those layers. So again, ecto, endo, and mesoderm. So the easiest one for us to identify is going to be the mesoderm, M for middle. So I'm going to start by annotating our diagram here with mesoderm being that embryonic layer in the middle. Up next is the ectoderm. Now the ectoderm is the external or the outside layer, and it's what's represented in blue on this diagram here. Lastly, we have what represents the inner layer of this, and this is known as the endoderm. Now, each of these layers will develop into slightly different things, and we'll go through what they each develop into now. So this first layer, the ectoderm, the outside or external layer, it leads to the development of things like the skin, the nervous system, the hair, and the nails. Now, you're probably thinking ectoderm on the outside forming the nervous system, which is on the inside. It's because of a series of folding that this can be found on the inside. These layers fold around one another in the development of the embryo. The next layer, the mesoderm, is the middle layer. And in the middle layer, the mesoderm will end up forming things like our 3 muscle types, so skeletal, smooth, and cardiac. It will also form things like cartilage, bone, blood. So think all of the connective, or most of the connective tissues found in the body. And it will also form some of the stomach and intestines. The last layer, the endoderm. This is the innermost layer, and it forms the lining of the alimentary canal and the respiratory structures. So things like the lungs, the lining of the lungs. It also forms a number of glands around the body and the urinary system. So these are the key things that I would know about each layer in terms of what they develop into. However, as we can see based on the textbook, there are plenty more for you to have a look at. So again, I would keep it short, sharp, and interesting with the few key structures that we've been through for each of these 3 layers. But if you really want to extend your understanding about these 3 layers and what they develop into, feel free to consult this table.